Hello, good afternoon, and good morning. My name is Yan Liu. I'm the moderator of today's webinar. We're very happy today to bring you a very interesting topic today. We will tell you how to close the gaps, what's the strategies for resolving imaging discrepancies in oncology drug trials. We're lucky we have very experienced three experts and speakers today, and I will make an introduction of them. So Professor and Dr. Navarra is a medical doctor and also full professor of pharmacology since 2022 at Catholic University Medical School, Rome, Italy. Currently serves as head and also chair of pharmacology, Department of Healthcare Surveillance and Bioethics. Dr. Navarra also has been in the advisory board for the Italian Ministry of Health, where he coordinated the Pharmacovigilance Committee and involved in the assessment of marketing authorization dossier and the national and also EMA procedures committee. We have our very honored speaker, Dr. Rojo, is MD and also PhD. She is a researcher in pharmacology, Department of Healthcare Surveillance and Bioethics, Section of Pharmacology, Catholic University Medical School, Rome, Italy. She is also a senior research associate in the Department of Pharmacology and Therapeutics Institution of System Molecular and Integrative Biology of the University of Liverpool, UK. Her research interests focus on the study of molecular mechanisms underlying neurodegenerative disease, such as multiple sclerosis and brain tumor. And recently, she has also been collaborating to research projects aiming to assess the extent of variabilities in evaluation of variants, cancer outcomes, and also its impact on the drug development. We have our last speaker, Dr. Antoine Yanissi. Antoine is our Senior Medical Director of Media and Technologies. He's also a very experienced radiologist, has more than 15 years of academic and clinical research experience, and was also the former head of Intervention Oncology Department of Cancer Center Nice Unicancer. And his research topic and interest is in the oncology biomarker development and also AI. Maybe just some words about the media and technologies before we start the topic. So media and technologies is a leading AI-based next generation precision medicine company. We focus in helping conquer cancer and other chronic disease through routine images and also through our imaging services for drug development. With our office based in the US, Europe, and China, we operate globally to deliver the best-in-class and end-to-end imaging service for your drug development. So today, we'll have very interesting topics. What you will learn in the coming 30 to 45 minutes. We will introduce you the impact of the site versus center read discrepancies on imaging outcomes and also drug efficiency evaluation. And what's the factors, root cause that contribute to those discrepancies? And then we will warm up and conclude with the ways to address and overcome those discrepancies. So now we will very welcome our first speaker, Dr. De Rojo. And I also a kind reminder that you can also submit your questions during the talk through our question panel, and we will have a very interesting roundtable discussions by the end of this webinar. So thank you very much. Now the floor is yours, Dr. De Rojo, please.
Thank you, Jan, for this nice presentation and for the possibility to present our analysis on this interesting topic. So, as you probably know, despite the overall survival is still considered the gold standard endpoint to measure drug efficacy in most types of tumors, An increasing number of drugs gained market authorization based on the evaluation of endpoints that rely on the analysis of radiological imaging. However, the main regulatory bodies will recommend the adoption of blinded independent central review for all clinical endpoints based on tumor assessment. These are including, therefore, objective response rate and progression-free survival. There has been also an ongoing debate on the added value of BICR to assess efficacy of new oncological drugs, and particularly the DODE collaborators first raised the issue of BICR as an unnecessary, expensive, and time-consuming procedure. And this was based on the analysis of 7 trials that did not show any significant discrepancy on the treatment evaluation among the 2 evaluators. And this was then confirmed by a larger analysis carried out by a pharmaceutical consortium of different pharmaceutical companies on 27 phase 3 trials, having the dual evaluation of progression-free survival. They found a strong correlation between the 2 assessments. We carried out our own personal analysis, looking for clinical trials that have the progression-free survival evaluated by local investigators and independent reviewers, and these were phase 3 trials. We searched the clinicaltrials.gov database, and we found 28 phase 3 trials with both evaluations. And as these trials normally have a control group and a therapeutic group, we could the data is always expressed as hazard ratios. And within these 28 phase 3 trials, we find a total number of 32 hazard ratios available for comparison. This data set is largely independent from the one analyzed by Amit collaborators, the paper that I showed you before, because there is only an overlap of 3 trials. Here you can see the data that we extracted from the database, and briefly we can say that most of the trials that we analyzed were in the setting of non-small cell lung cancer, renal cell carcinoma, breast cancer, and in the oncohematological setting. And the vast majority of these trials, 68% of the trials, had an open label design, therefore requiring a blinded independent central review. We extracted from the database the hazard ratios as it was provided by the local investigator, as well as the hazard ratio provided by the independent assessors. And we calculate what we call the discrepancy index, which is a ratio of the hazard ratio of the investigator versus the independent reviewers. With a null hypothesis of no difference, we were expecting an hazard ratio discrepancy index of 1 for those trials that had no difference. And you can see here there are 2 trials. An hazard discrepancy index above 1 for trials that have the blinder investigators more optimistic than the local investigators, and discrepancy index below 1 for those trials in which the local investigators were more optimistic in the evaluation in comparison to the central readers. However, in the pooled analysis of these 32 hazard ratio comparisons, we found a mean discrepancy index of 0.98 with a 95% confidence interval, including the value of 1, therefore suggesting a complete concordance between the two types of evaluation. We ran also a subgroup analysis, and we found, for example, that for trials carried out between 2003 and 2006, there was a trend. Actually, there was a significant overestimation of the effect by the blinded assessors, but this was not happening in the following years. We found a larger variability between 2013 and 2016, sorry, due to the inclusion of 3 studies that had the local investigators more optimistic versus the blinded investigator, and this was largely due to the Keynote 61 study that evaluated the efficacy of pembrolizumab, which also explained the larger variability that we found looking at biologicals versus small molecule drugs. So overall, this data, in line with the current with the evidence from the literature, demonstrate some variability, but no evidence of systematic bias between the two assessments. And this also explains the publication of several recommendation papers that suggest limiting the blinded independent central review to those high-risk situations, also considering the current trend to include the data of real-world evidence. BICR may appear as a source of bias, being distant from what we normally do in clinical practice. On the other hand, in phase 2 trials, we often found another clinical endpoint for the evaluation of drug efficacy, which is the objective response rate. This is always chosen as primary endpoint in early phase, and an increasing number of drugs gained conditional approval based on objective response rate. So we then tried to see whether there is a discrepancy between local investigators and blinded assessor with for the evaluation of this clinical endpoint. And here you can see our search strategy. We identified 20 trials, including a dual evaluation of objective response rate, 10 of which had also a dual evaluation of progression-free survival. These trials were mostly in the setting of non-small cell lung cancer and in the oncohematological setting. All of them had an open label design, and 60% of these trials had a single group assignment, which means that most of the trials lacked a comparator arm. Here you can see the data that we extracted from the database, and we calculate a discrepancy index for the objective response rate and from the progression-free survival. However, here we use the median time for the calculation of the progression-free survival discrepancy index. And with an index of above 1, meaning that the local investigators were more optimistic, and a discrepancy index below 1, meaning that the blinded assessors were more optimistic. Here are the main results. As you can see, based on the mean discrepancy index, we can see that there is an overestimation of the objective response rate by 17.5%, whereas there are no difference in the evaluation of progression-free survival in phase 2 clinical trials. the trend to overestimate the objective response rate and therefore drug efficacy by local investigators also appears in the subgroup analysis, and we can see that there is this effect also over the years. We did not find a significant correlation between progression-free survival discrepancy index and overall response rate. And the conclusion of this analysis was that we can see that there is a clear overestimation of the objective response rate, therefore drug efficacy by local investigators in phase 2 clinical trials, whereas there is a substantial agreement among the two evaluations for the progression-free survival. In the attempt to understand better these results, we ran a final studies in which we look at phase 2 and phase 3 trials, having both objective response rate and progression-free survival with the dual evaluations. And here is the research strategy that we adopted. We selected 17 phase 2 trials and 21 phase 3 trials for this analysis. Here is again the results that we extracted from the database to calculate a discrepancy index for both the clinical endpoints, and this is for the phase 3 trials. Very briefly, for the phase 2 trials, we found a substantially similar result with our previous analysis. So most of the trials were in non-small cell lung cancer and non-hematological setting. All the trials had an open label design and lacked a comparator arm. And out of 31 comparisons, we found that there was a significant overestimation of the objective response rate in using these larger samples that also included 9 of previous studies. We found a 24% of overestimation of the objective response rate, whereas there was a substantial agreement for the progression-free survival. And this was in line with the first analysis. In phase 3 trials, here you can see that trials are more scattered toward different therapeutic areas. 81% of these trials had an open label design, and approximately 50% of these trials had progression-free survival per central reviewers as a primary endpoint. And out of 42 comparisons, we could still see this overestimation of objective response rate by the local investigators, whereas there was complete overlap for the progression-free survival between the two evaluations. However, since this trend to overestimate the objective response rate occurs both in the control group and in therapeutic groups, when we look at the ratio of the odds ratios between local investigators and independent central review, even though this was limited to only 4 trials, we did not find any significant discrepancy in the overall estimate of drug effect. And this is in line with the previous large meta-analysis of 76 phase 3 randomized controlled trials, including more than 40,000 patients, in which these authors they evaluate different clinical endpoints. They found some trials with significant variability, 8 trials regarding the objective response rate, 10 trials for the progression-free survival. But in the pooled analysis of all the trials, they found a substantial agreement between the local investigators and the blinded reviewers for the evaluation of objective response rate and progression-free survival. And this is also has been recently demonstrated by a large meta-analysis carried out by Roche investigators on 49 trials The only difference is that the analysis included both the early phase and the phase 3 trials, and they demonstrate a substantial agreement between the two evaluations for objective response rate and progression-free survival. So in conclusion, our data and data in the literature suggest that while for the progression-free survival, there is a substantial agreement between the local investigators and the blinded reviewers, for the objective response rate, there is this significant overestimation by the local investigators, and this is particularly relevant for non-controlled clinical trials, so those early phase trials that lack a comparator arm. This can also impact on approval, drug approval, as well as a future program of drug development. Thank you all for your attention, and I will now pass the microphone to Dr. Iannassi for the next part of this webinar.
Thank you, Dr. Del Russo, for this comprehensive analysis. On my side, I will continue going into more specific cases and trying to explore the root cause analysis of variability behind local assessment and independent central review assessments. So initially, I need to give a larger perspective on centralized analysis imaging. Usually, everyone knows that patients included on site, they have a first review from the radiologist, investigator radiologist. So patients have their image acquired, then analyzed. And those images are centralized through an imaging core lab. After a first quality control, those assessments will be provided to independent readers, so one or several independent readers, and it is actually possible already to have discrepancies between central readers. But usually in this case, these discrepancies are solved by a planned paradigm of reading, including usually a third reader known as an adjudicator, deciding which one of the two central readers will be kept for the endorsed result. So here we can have discrepancy, but the topic of today is actually about a different type of discrepancy between the result of the investigator site and the central assessment. In terms of variability and discrepancy, we can observe variability on patient eligibility to the trial at baseline, but also during drug efficacy assessments for the assessment of the trial endpoint, response, usually for phase 2, progression, usually for phase 3. So the remaining part of this webinar, I will use basically an agenda in regards to the three sources of variability. The first source of variability that we can discuss is the information itself. As you know, those images are provided as a source images to central readers. So we can discuss why those assessments are different based on the same information. And we will see that actually the information can be the same, but the availability of contextual information might be different. The second reason or driver of variability can be linked directly to radiologists. So the observers have different expertise, but not only the expertise, the condition of work are playing a role in terms of variability. And the third reason of variability might be linked to the rules of assessment. So to assess oncologic images, we use criteria such as the RECIST criteria. And here also we will see that some subjective parts are included on those criteria and might be a reason for variability. So starting with information, it might be not easy to understand why one same image might be interpreted differently between the site and the central assessment. So let's see how those information are actually assessed on site. So the radiologists on site have full access of the patient medical record. That means that he has access to the previous images of the patient, previous treatment usually, and he can discuss verbally of those cases with the colleague on site. So that's the condition of information assessment on site. At the opposite, when we centralize those information, the information that is centralized is set as controlled information. So only one part of the information will be centralized, and especially for clinical information, those information will be decided and planned before the assessment, and only some specific information will be provided to the central readers. Usually, for oncologic assessment, we provide prior treatment for the baseline analysis, such as radiation therapy, to avoid selecting an active lesion. And during treatment, it is possible to also provide some concomitant therapy or some histology report to those readers. But the access of the information is restricted and predefined during the trial protocol and reading rules. So to give an example of what can be the influence of controlling information, it's better to give actually to use an example to make everyone understand. So I provide you here one image, and I might ask you in front of this image, what do you see? So if I ask this question, probably one part of you will tell me that I see a duck, and another part of you might see maybe a squirrel. None of you are actually wrong, because the information, the data is actually ambiguous here. But if I provide you context to this image by adding a point, I ask again the question here, I will expect most of you to tell me that they see a duck. And I will probably have this result if I do a poll. So this explains why when we control the information and we restrict it, the assessment might be different between radiologists in front of the exact same pixel information. This has been proved. Some authors have been reporting what is the influence of clinical information in addition to pixel information, and they demonstrate that it improves the interpretation accuracy, the clinical relevance, and also the reporting confidence. So speaking about confidence, there's one key word to keep in mind is the equivocality. Indeed, when we centralize images, there are some cases that might be equivocal. Here I provide one example of one patient for lung cancer, centrally assessed with targeted 2 lesions. One of the lesions was selected in the adrenal gland, the other one in the lung, and those lesions were followed. The outcome of the assessment was stable disease. Locally, the local investigator had one additional information in regard to the adrenal gland lesion that actually, when you look into the assessment, remains stable, unlike the second targeted lesion. So having this information, the local assessor did not take the adrenal gland, and the outcome it was different, having a patient responder on site and a non-responder centrally. It gives you an idea on how having information, especially for benign pre-existing lesion, it can influence and explain part of the discrepancy between site and local assessment. We actually carried out one more quantified simulation about what is the impact of those, including equivocal lesions at the baseline, and we demonstrate that it can the threshold used for clinical assessment, for radiological assessment, to assess the progression or the response, which is normally 20%. But if we include some equivocal lesion at baseline, those thresholds are shift, and they can explain discrepancies between readers and assessments. During follow-up as well, there are some equivocal situations, and here the main cause for equivocality during follow-up are linked to adverse events or intercurrent inflammation, which is on site easily analyzed due to the clinical situation, which is known, while centrally sometimes it can be difficult. Here, one case of one head and neck cancer. At baseline, there was no bone lesion in the jaw, but at the 17th time point, slowly one lesion, lytic lesion, appears in the bone. So in regard to this type of lesion, one radiologist will probably propose two different differential diagnoses. One is a new bone metastasis, but one will also think about mandibular osteonecrosis. Centrally, the radiologist will have to make a decision between progression or complete response, knowing that all the target lesion had disappeared for this patient. So without any additional information, there is one chance that it can be PD or it can be a complete response. But on site, the radiologist had access to the patient history and knew that this patient had received bisphosphonates. So in this case, of course, the most probable diagnostic here is the mandibular osteonecrosis. So to give you really an idea why clinical information might impact the outcome of the assessment. The second reason for variability might be linked to the observer himself, and indeed, on site, it's important to understand that the radiologists have a very high workload. They have heterogeneous experience on oncological images. They don't have a dedicated system for response assessment usually, and also it's not always the same radiologist that reads the same patient images. There is also difficulties to have lesion-based traceability for site analysis, while when we centralize images, we use a certified radiologist, which is specialized in the field and the indication of the trial. We know well the criteria, so the radiologists know well the criteria with most probably previous trial experience and pitfalls that have been experienced by the reader. There is a same reading system for all the readers, which include some lesion tracking, automatized, and compliance check. And it's always the same reader that will read all time point, all visit examination for one same patient. So it here what we perform for centralization is basically we try to control the variability using tools. We also monitor this variability at each step of the trial, and we can expect more variability between site and central reading than the central readers. To probably give you more an illustration about what speaking about in terms of really information, the way we report images on site usually is reported by using paper, and there is no electronic case report for images assessment. So this is the most common way radiologists analyze on site, and information is gathered to the ECRF, while centrally the system is automatized between the information at the pixel level and the information translated into a report. So we, in 2019, carry out one studies to quantify what are the uncompliancy measured between the site and the central assessment, and we noticed that some of those uncompliancy, such as selecting non-RECIST targets or selecting the wrong time point as a nadir, or having an erroneous sum, for example, those uncompliancy, they can explain part of the discrepancy between the central reading and the site reading. In fact, centrally, those they are usually avoided by the use of dedicated tools that are not always available on site. This said, it's not so easy to integrate those tools on site due to the diversity of the IT system, and so it's quite challenging to provide for site such tools. The last reason that the last, let's say, root cause that we need to analyze is the rules of assessment. So when we provide the cases to the radiologist, together with those cases, the radiologist will use criteria to assess. Usually all criteria are more or less based on the same type of assessment. One part is quantitative, one part is qualitative. Why all criteria are not quantitative and objective? It's because we need to have one rule of assessment that balance the fee and I would say the efficiency of the assessment. So you will always have one part of the disease that cannot be quantified objectively, also because one part might be just not measurable, for example. This part is housed into what we call non-target lesion, and new lesion will be assessed during follow-up. Those parts as well, usually they are not quantified. This said, if we look more specifically into the RECIST assessment category, there are some factors of variability, and some of them are totally subjective, even though they belong to a category of lesion that are measured. For example, the target lesion, they are usually measured for each time point, so one can consider that they are objective, their assessment is fully objective. Actually, it's not the case, because the selection can be different between readers, but also in terms of disappearance of small lesion versus scar, for example, this is subjective. And for other categories such as non-target, we have variability linked to a subjective opinion when applying those rules of analysis. So we, in 2023, so lately published a paper analyzing 1,720 lung cancer patient assessment. So it was a double assessment. And when we look to that, at baseline, we can provide some figures of variability. So looking specifically to eligibility, the rate of discrepancy between the measurability of the disease at baseline versus non-measurable patient was very low. So meaning that there is actually low discrepancy between the central assessment and the site assessment in terms of eligibility, about 5% generally for those type of indication. When we looked into radiologists assessing those patients, and here we had about 10 radiologists assessing those patients, we have noticed that in terms of baseline analysis, the definition of the disease at baseline is variable. So one radiologist can see at baseline a distribution of disease which is different. Some radiologists might consider that there is a bone metastasis, when another one will not consider that there is a bone metastasis. Same for lung, same for node or pleural lesion. Also, when looking at one readers, we would notice that all radiologists have their own pattern of reading. So there is really some specificities which are linked to the observer in terms of assessing one same patient. So there is one part of the variability at baseline. And if we look to not patient, but one lesion, we can also explain why one lesion is assessed differently by two readers. This is one case of one lesion assessed by one central reader and a site reader. It can be linked to the measurability for small lesion who does not reach 1 centimeter. So one reader will say one will say it's not measurable. It can be linked to the localization of the disease. Here we have one reader for the same lesion that consider this mass as a lymph node. So the methodology of measurement is different and the assessment is different. And it can be linked to the measurement itself, the way we see the contour, the delineation of the disease, and there can be some quite significant variabilities between two readers. This is a real case of central versus investigator eligibility discrepancy. One case for lung cancer. At baseline, there is one lesion in the lung that actually diminished at the first visit, and then there is one mass at the second visit. Here it's hard to delimitate this lesion due to the lung atelectasis. Another case, here we have one lesion which is in the head and neck and the pharynx area. At baseline, the visit 1, visit 2, it's difficult to measure with accuracy this type of lesion. So centrally, the lesion was selected as non-target lesion by both central readers. The patient was said basically not eligible for assessing the response. Also, but on site, those lesions were probably selected as target lesions. One specific variability about PD confirmation. So this event can be sometimes centralized to have a centralized assessment. And here it's an example of 3 different assessments. The site asked for PD confirmation. The central reader provided also assessment at the time point 1, but there was a tendency to increase, but was not significant in terms of RECIST assessment. So here we have a variability. We see that the tendency is the same, but the outcome is different. Here another case, a bit different, where we have the site requesting confirmation of progression at TP1, TP2, TP3. We see that our central readers, they provide also assessment. There is an increase of progression. Only one reader say that there is progression at TP3, but see an adjudication is performed, and it's the central reader 1 without PD that is endorsed with an explanation of the adjudicator. So it's not only response, but progression can also be variable, and assessment can be quite different between readers. So in conclusion, here it's important to differentiate 3, I would say, items in terms of rationale for the blinded independent central review. The first one is the quality of data. So the FDA actually requests to have some high quality information for the trial assessment. The second one is to avoid bias, so using blinding and blinding information. And the third one is to control the variability And those three are actually needed to provide some correct assessment. So we can already say that depending on the context, it's difficult to generalize our conclusion, even if we will try, because of course, due to the role of the assessment, the drug type and the effect on the tumor, the study design, the criteria, and the disease presentation, it's actually sometimes it can be more keen to bias, sometimes more to viability. So it's hard to generalize conclusion. This said, what one needs to understand is that it's important to be aware that there is site versus central viability and bias. We have seen that bias are mostly observed in phase 2 single-arm trials, and there is still high viability between site and central readers for phase 2 and phase 3. It's important to ensure centralization and adequate information, which are contextual information, as we saw, such as clinical information, especially at baseline, to have initial disease definition, which is quite the same between site and central. It's important also to provide good training with challenging cases in order to harmonize the assessment of the trial at initiation and follow it up. Following monitoring the discrepancy rate and analyze the main discordance reason during the study. Sometimes it's important to retrain the readers or on site. And all this must be planned. So it's important to have a site versus central discrepancy management. And this management needs to keep the independency of the imaging central reader.
Oh, thank you. Thanks a lot. Thanks, Dr. De Rosso and Dr. Nisi for the excellent talks. And now we will enter into the panel discussion session with our three very experienced experts, Dr. and Professor Navarra, Dr. De Rosso, and Dr. Nisi. I see that we have already received several questions via our question panel. So I will read it out and I will also submit those questions to our experts. I see there was first one, probably for Dr. De Rosso, for you. It says, have you looked also at the BICR versus investigators read for neuro-oncology endpoints? For example, renal HGG, so high-grade glioma, and renal LGG, low-grade glioma. And I know that there were failed phase 3 trials, but curious to know if there are any concordance between BRC and investigator changes based on imaging modalities, because they are using, one using CT MRI based, the other is more brain MRI based. So, shall we start with Dr.
We haven't looked specifically to these neuro-oncology criteria in our analysis. What we can say that we had very few trials in the central nervous system setting. Like there was a very small phase 2 trials on medulloblastoma showing increased confirming this trend to overestimate the response, the objective response rate by local investigators. We had also a phase 3 trials in glioblastoma in our sample of trials. We did not observe any substantial difference for progression-free survival, but we haven't looked at these other comparisons. Interestingly, this trial had overall response as a primary endpoint. I hope this answered the question.
Thank you. Thank you. Okay. I have a second question, I guess, that probably will be for Professor Navarra, because you are expert also with the regulatories. So what is the regulatories' opinions and views on those discrepancies? Do you think there was any threshold saying, oh, this discrepancy is already 40, the trial will fail? So what will be your opinion on this?
Yeah, thank you for your question. You see, the regulatory bodies, they seems to have a different sensitivity to this problem from one side of the ocean and the other. I mean, we have seen that at the FDA, probably is more it takes more into account of the results that we've shown here, because not always there is not always a compelling request for BICR. In the US guidelines, it is stated, okay, it's based, it's a case by case based decision. So this leaves open a possibility to for instance, to choose a local investigator as a primary endpoint, local investigator assessment as a primary endpoints. In a way, it's closer to our result. This approach is closer to our result. On the other hand, we see that in Europe there seems to be a more conservative approach. One of the papers that have been shown by Cinzia Dell'Orusso is actually was published by a group of EMEA experts. They already about 10 years ago, they were in a way they were soliciting to change policy, to have a more open view about the possibility to have a local investigator as a primary endpoint. But so far it seems that there's been no change in this direction. Of course, this is a changing scenario, because as far as we have a larger number of trials to be analyzed, probably we will have more possibility to go into detail. But at this stage, what we can see probably is that there may be there is no doubt that local investigator and big assessment, they are interchangeable in a way. There is no statistical difference, so there may be no need to for a blinded review in a phase 3 trial having a PFS as a primary endpoint. Seems that we may be relaxed about this setting. On the other hand, some concerns still exist as far as overall response rate are concerned, especially if the trial is designed with a single group study. Because in this case, we have seen that there is this variability. It seems to be related to a bias anyway, because local investigators, they tend to overestimate their observation, either in the case of experimental treatments and in the case of control treatments. If the study is designed in order to have a control group, this bias is counterbalanced by the presence of a control group. So actually, the only major consensus is now related to the case of single group studies having overall response rate as a primary endpoint. So this is trying to summarize what we know as today. Of course, it's very interesting. I also would like to add that the approach that has been shown by Antoine Yanesi to go into the detail at patient level is very helpful. Nevertheless, it does not explain yet why we have this discrepancy, because the kind of analysis is the same for a PFS and overall response rate in a way. is based on imaging analysis. And still, we don't know why there is some discrepancy when we look at overall response rate, and this discrepancy is not present when we look at PFS. This is still a major question mark at this day.
Yeah, that makes a lot of sense. And this is also our impression that regulators, they really look into many experts, many dimensions, what the disease indication, how is the discrepancy, whether the data are reliable, is there any bias? Indeed, there was no one size fits all. Yeah, exactly. I see that we have more and more questions. So the other one will be, have you considered skin cancer and the impact of dermatology reads into this outcome of objective response rate? So who will start? Maybe Dr. Yannissi, you want to start this question?
Thank you, Ian. So I think here we have several questions that actually in line on what we just mentioned. In terms of indication, I believe that the indication and generalizing the let's say our conclusion to all phase 3 or all phase 2 or one endpoint or one or PFS or I think it's quite difficult. And for dermatology, for example, indeed, if one looked specifically to dermatology, it can't be fully comparable to radiology assessment. We need to have a specific and dedicated analysis on those indications. So actually, I'm not able to answer. We have not, on my side, I've not specifically looked into those indications, but I can say for sure that it would not be we need to specifically look into this case, such as on my example, for example, I shown some patient with head and neck cancer, and head and neck images and presentation of this disease is quite variable. Not the same variability than lung cancer, and we can expect that site evaluation, central evaluation in respect to different indications, skin cancer, head and neck, lung, they are not the same. We can't really say that the accuracy can be the same with, as we explained, for all the contributors of this variability. Just for example, the expertise of radiologists or the use of different radiologists. So bias is one thing to consider, but quality and accuracy of data is also part of what needs to be considered here. Thank you for this question, because indeed, I think dermatology or some specific indication, neurology, needs to be analyzed separately, for sure. Yeah.
Yeah, indeed. For skin cancers, they may have a more complex design and integrate those clinical lesions into the response assessment. And I think Median has also experience to handle this and also drive those trials to regulatory approval. Indeed, the reading diagram are more complex than just a simple solid tumor like lung cancer or breast cancer. Yeah, we may also think to do further on these topics. Yeah, because today I think we are also short of time. We have maybe time for last question. Is are there drug approvals that have been given based on local reading alone without blinded independent central reading? I think this is a question again for all the readers. So, so who wants to start for the last question?
Well, in our analysis on phase 3 trials, we found, for example, that the progression-free survival was in a limited number of trials. The progression-free survival by local investigator was set as the primary outcome of the trial. And these regard 4 open label trials sponsored by Roche, 2 in non-small cell lung cancer and in the oncohematological settings, which again, if the trial is well carried out, the local investigators has enough the ability to provide reliable results. There were also 2 other trials that the local investigator progression-free survival set as primary endpoint, but these were double-blinded trials, so which are in the case of what Professor Navarra just said, that if the trial is double-blinded, maybe the regulator accept more this kind of approach.
It's also not uncommon to have, especially for phase 3, to have several endpoints. Some of the endpoints are actually assessed locally, and it could be that for some indication, I'm thinking about neuroradiology and RANO assessment for PFS, for example. Sometimes they are assessed locally, but if you look into the protocol, you will see that several endpoints are usually also included as a secondary endpoint, or and those needs central assessment.
The majority of these trials had the blinded review as primary endpoint for progression-free survival, these phase 3 trials. There were few trials having both as primary endpoint, and there were very few having the local investigators, which are the one that I just mentioned. So it seems that the regulatory still rely more on blinded independent central review. There is, although, a trend to include the local investigators towards the most recent trials.
Yeah.
But if I may add, Cinzia showed a paper by the first author was EMIT. That was a consortium of companies that discussed their own data, and it was about 10 years ago. So what I can see is that the group of companies that were part of this consortium, later on they are a bit more, let's say, propositive on this side, because Roche was one of them. So this seems that if the companies that are more sensitive to this kind of problem, they analyze their own data, in this case they are even more confident to present data with the local investigator assessment as a primary endpoints. This seems that in this case the companies are trying to lead toward a change in the regulatory asset.
Yeah,
that we see.
Yeah, indeed. But this is a very, very good topic. I think regulatory will always think about potential bias, variability, and quality traceability. And also take into account the cost of drug development. I think this maybe deserves another topic that we can dig into. And today, I think I would like also thanks to 3 of you. Thank you very much for your time, and also our audience for your questions and very interactive discussions. So for those, if you have also the questions we're not able to answer now, we will answer that question to you by email. So don't worry about that. We will contact you by email. And also, if you want to read and also listen to this webinar again, the replay will be available at our website and also at our LinkedIn very soon in the coming weeks. So thanks a lot, and I hope to see you next time. Thank you.
Thank you.
Thank you.
Thank you.